Next up we have Avi Mayan from Icahn School of Medicine at Mount Sinai. He's going to be speaking on GENIVA, aggregation and analysis of gene expression signatures from related studies. There we go.
Hi. So GENIVA is a new tool that we've developed in the lab. And the purpose of this tool is to create collections of gene expression signatures and then analyze those collections using interactive reports. So in order to understand GENIVA, or GENIVA in general, it's an expansion of a project that we published last year called GEO2Enricher. GEO2Enricher is a Chrome extension. It's a piece of software that you can add to your browser by going into the Google Play Store. And then nothing really happens until you go to a GEO page. And this is, for example, a data set landing page at GEO that what we want to do in this data page, we want to add functionality to this page. So a Chrome extension can hijack the JavaScript and HTML code of a website and add functionality to a website. So after you click this button, compare two set of samples, there is an appearance of this blue link that is not there when you don't have the extension installed. But before you actually click on that link, you want to select your samples. You want to select your control and your experimental treatment samples that you want to create a signature for. In this particular example, we are looking at study that looked at the changes in expression of skeletal muscle of mice over age. So looking at young versus old mice. So there is 2 months old mice and 5 samples, as those going to be our control samples, or the ones that are on the left group. And then we have an aged mouse, which is a 2-year-old mouse. And there are also 5 samples that we selected. And then we click on that blue link, and this pop-up dialog box appears. And that pop-up also allows us to add additional metadata about our signature. And most importantly, you can add a metadata tag, which is a hashtag, that sets up the ability to extract signatures into a collection for the reports, for the GENIVA reports that I'm going to talk about. So here the hashtag is aging_BD2K_ all hands meeting _demo, and that hashtag will be stored with the signature in the database, and if we extract many of those signatures, we can generate a collection, and then that collection be analyzed together. So this is the results page. So after I submit the information from the form to our server, we compute the signature, and then we store the signature into the database with the tag. And this is not the same hashtag that I showed. This is a hashtag that we created for collecting gene expression signatures for aging using a crowdsourcing project. We recruited students from a MOOC that we are running on Coursera to extract signatures, and the instructions was, it doesn't matter if it's like mouse, human, or rat, and the tissue also doesn't matter, but trying to compare tissue from a mature adult that is young versus elderly, either mouse, human, or rat, of the same tissue. And the crowd collectively generated 245 signatures from different studies that they identified from GEO. And now, automatically, we're generating this interactive web-based report. And the first part of the report is a PCA plot that cluster all of the signatures, all those 245 signatures, to see how they spread and how similar and different they are across different tissues, and the coloring is by tissue. This is an interactive map that you can rotate. The next interactive visualization in that report is a clustergram or a heat map, and those are the 20 genes that are the most consistently upregulated or downregulated across all those signatures. You can also interact with this heat map, and you can make it, if you want to look at the top 100 genes, or if you want to sort the genes by the most differentially expressed, or you want to filter by different mechanisms, you can do that. And this is implemented using a tool that we are developing called Clustergrammer, and there is a poster that is just described this tool next door. The next analysis is enrichment analysis vectors, and this is the GENIVA name comes from that type of unique analysis. So what we do here, we compute enrichment for the up and down genes for each signature, and then we cluster the enriched terms. And in this particular example, we do enrichment analysis against the ENCODE data, or the data set from ENCODE, and what we see is that many of the regulated genes across those 245 aging signatures are regulated by NF-kappa B or REL-A, which is probably indicating of signal of inflammatory genes that are upregulated. The final part of those interactive reports is predictions of drugs or small molecules from the links L1000 data that can either reverse or mimic the expression across all of those signatures that are most consistently predicted to reverse expression across all of those signatures. And here we find some natural compounds that are profiled by links that are predicted consistently to reverse these aging signatures that I showed. This is another collection, and looking at the drug analysis, the last part of the analysis for dexamethasone. So we extracted 86 signatures from GEO for this dexamethasone. It's a glucocorticoid steroid that is an anti-inflammatory drug that is used for many diseases. And what we see is that 1/3 of the, how do I go back? 1/3 of the signatures are also recovering L1000 signatures that profile dexamethasone. So this indicates that regardless of the lab, the cell type, the organisms, we get the same genes that are upregulated by dexamethasone and are predicted to be mimickers of dexamethasone. So dexamethasone is selected out of the 20,000 small molecule drugs that are profiled by links to be the most similar to those signatures that we found in GEO. And then we also find additional drugs that no one really knows what they do and have a very similar pattern as what dexamethasone is doing, and those could potentially be useful leads for having some functions for those drugs as potential anti-inflammatory drugs or a substitute for dexamethasone, which causes a lot of severe adverse side effects. So a little bit about statistics about what currently we have in GENIVA. We have 22,000 signatures and about 300 of those hashtags and 190 reports that, like the report that I showed you for the aging. The paper was published 2 weeks ago in BMC Bioinformatics, and currently it only supports microarray data, 70 different microarray platforms that are on GEO. And what we are trying to do now is we try to do it for RNA-seq. So Alex Lackman, who is an instructor in our center, he already, using the common credits model, aligned all of the RNA samples for human, mouse, and rat from GEO and SRA, which is about 180,000 samples, using some cloud computing. And this is showing the quality of aligned reads. So on the y-axis is the percent of reads that were aligned, and the institution or the core facilities in institutions across the world. And we can see that there is a high variability in the quality of those core facilities across the country and the world. And so this is a poster also that you can attend after these talks. So this project is work of a lot of people that contributed different parts. Gregory Gunderson developed both the GeoAutoEnricher and the GENIVA tools. Nicholas is here and he developed the clustergrammer tool. Zichen developed the crowdsourcing component of this project. Max developed the Enricher APIs. Alex and Dennis are developing now this RNA-seq support for this app, and Troy and Caroline are extracting RNA-seq signatures for the future part of this project. So I just would like to acknowledge the grant support from the NIH, and I think I made it in time. All right. Thank you.